So thank you for the introduction, and thank you to Hassan, Baldomir, and Frederick for the invitation. I guess I've been doing neuroergonomics for a while. I started out as a graduate student many years ago, 1979, and I had two mentors. Well, I had five, but I started out with two. And one of them was Manny Donshin. Some of you know that name if you do any work with the P300. And the other one was Chris Wickens, very well known in human factors and engineering psychology. And at the time, Manny and Chris were putting together, I guess what today we'd call neuroergonomics. At the time, it was biological psychology or cognitive neuroscience. The terms have changed over the years. So my research over the years is focused on a number of topics, from using ERPs and EEG and eye tracking and many other technologies to study mental workload, attention, training, both in the lab, in high resolution simulations, including VR, and in the real world. I remember building a transmitter in 1983 to telemeter some EEG and heart rate data from a high-end simulator about a block away into a control room. And somehow we built it with Radio Shack parts sitting in a hotel room the night before. And it actually worked. Amazing. These days we have much better systems, of course, and many of you are developing them. As a result of the research I've done over the years with my colleagues and students, I got particularly interested in lifestyle and the decisions we make well as humans, but we make a lot of decisions poorly about physical activity, diet, intellectual engagement, social interaction. So the kinds of things I do, illustrated by some of the cartoons here, I think cut across a number of the topic areas in neuroergonomics. But what I'd like to tell you about today is some work we've done over the years on physical activity and exercise, illustrated by a little age-defying fitness there. So the kind of work I do is highly interdisciplinary. In fact, I consider myself as being interdisciplinary for the last 35 years. Before coming to Northeastern, I was director of the Beckman Institute for Advanced Science and Technology that has 235 faculty and 1,500 researchers, all of whom are interdisciplinary. So these are some of my colleagues across the world who I've collaborated with and do collaborate with on this research. A lot of the inspiration for what we do is the animal literature, and a particular colleague who's passed away a few years ago, his name is Bill Greeno, who started some of the original work on neuroplasticity with rodents. And we have a photograph on the top left of Bill's lab in which the rodents are exploring new toys or stimuli. There is a Cirque du Soleil, this is for Frederick, I think, since Cirque du Soleil came from France, animal who's learning the trapeze there. Bottom left illustrates some of Elizabeth Gould's work on social interaction and how it impacts the brain. And we'll talk about the little rodent on the right here. They don't usually use treadmills, running wheels, but nonetheless. But this research was quite interesting, and I'll show you some of the molecular and cellular effects in a second. But another inspiration for the kinds of things we've done is the lack of physical fitness. I used to think this was a uniquely American thing. I work with some colleagues in Spain. I can say it is not, that this is a problem throughout the world. But, on the top photograph, the little doggy is getting exercise, the gentleman in the truck, not so much. And we wouldn't want too much exercise, so we need those escalators to get up to the fitness club. So sedentariness, obesity have become huge problems in our industrialized world. These are some of the topics I'll talk about today, and very briefly. And if anyone's interested in more or has any questions or any suggestions or comments, I had my email on the first slide. I'm at Northeastern. I'm also still have an appointment at the University of Illinois. You can find my email in either location. I get both of them. But what I'll start off with is to get a little bit into the animal research, because it provides the cellular and molecular underpinnings of what I'm going to talk about with humans. My work is mostly with humans. And this is a slide from a colleague and friend, Henriette von Praag, who was at the National Institutes of Health for many years and is now at a university in Florida. And it illustrates the kinds of research that are done. They're really quite straightforward, mostly interventions, some cross-sectional research. And with the rodent research, there's either a running wheel or not. You have the little hypodermic needle on the left-hand side. That's an injection of a substance that'll track cell division, be it neural or otherwise, called BRDU. There's usually some learning situation, the Morris water maze, in which you put the rodent in a opaque bath of fluid, of water, and it has to find its way to the platform because rodents don't like to drown any more than we do. And then some measures, electrophysiological, histological, and so forth. And that's the way most of these experiments work with rodents and with humans. We don't throw the humans into the bath of water, but other than that, it's pretty similar. And we don't do the brain histology. But here are some of the results from a literature that dates at least 20 years, probably more like 30 years nowadays. And the photomicrographs on the top left are from the hippocampus. Those little green dots are actually new neurons being born in middle-aged rodents. A middle-aged mouse would be about a year, a little over a year of age. And what's been found in this literature is that these new neurons are born in the hippocampus throughout the lifespan as a function of physical activity. And the bar graph, these are some data from Rusty Gage's lab at UCSD, indicates three conditions: control, enriched environment, that's with all the toys and stimuli, and the running. And we have an illustration of the number of new neurons that are born. But if it was just new neurons that are born selectively in one or two regions of brain, not everywhere, that would be interesting in and of itself. But there really are a number of molecular and cellular changes that occur, some of which we can tap in humans, perhaps not in exactly the same tissue that we'd like to get the information. We don't do biopsies of the hippocampus or other regions of brain too often in humans, but increases in neurotrophins, which are nerve growth factors, increased synaptogenesis, or connections between neurons, angiogenesis, which is vascular structure, and a variety of other changes listed here. A little bit further down, I've listed reduced beta amyloid protein in transgene mouse models. Well, that's particularly interesting to me, and I'm sure to many others, because beta amyloid is one of the protein precursors for the pathology, the pathology in Alzheimer's disease. And what's been found in these transgene mouse models is that amyloid and tau, another protein that mucks up the brain with Alzheimer's disease, tends to be downregulated as a function of exercise. And as a function of the downregulation of these proteins, you have an increase in learning and memory, which is really pretty interesting. So perhaps exercise is one way to slow down the process of some age-associated neurodegenerative diseases. Okay. Moving on here, I wanted to talk a little bit about meta-analyses. Many of you, I'm sure, have done them or have an interest in doing them. They're, as I tell my students, I'm working with a couple of students on one now, the math is trivial. The hard part, the math is simply algebra. The hard part here is selecting the right studies with your inclusion and exclusion criteria and selecting the right moderators. But there have now been dozens of meta-analytic studies attempting to summarize the literature, and it's a great way to do that and to point out the gaps, what we know and what we don't know. And here's one that Stan Kocham and I did a number of years ago and updated a couple of years ago, and just one plot. The simple question was, does exercise improve cognition? No physiology here yet. There'll be plenty of physiology later. And the answer is yes, it does. And one of the moderators is or are the nature of cognition. So you get the biggest effects, the unfilled bars are from those humans, older humans who participate in aerobic exercise programs, could be bicycling, swimming, walking, jogging, running. You get the biggest effect compared to the control groups. These are from all randomized controlled trials in executive control processes, and it tends to decrease as we go from left to right. But the interesting aspect of this is we tend to get benefits for exercise across the board. So you might ask, so what's happened in all these years? Because this is an older meta-analysis. And here's one from a colleague at UBC, Teresa Lou Ambrose. And I've just summarized it, but it's published just, I don't know, 7-8 months ago now in the Neurobiology of Aging. And the bottom line here are the last 2 points under the highlights. exercise training improves both physical function. Teresa and her students looked at physical function and cognitive function, and the effect size is small to moderate. And there's a relationship, that is a correlation, if I remember correctly, of about 0.45 or something between physical and cognitive. So as people become more fit, their cognition improves, or perhaps vice versa. It's often hard to know. So this is a very recent meta-analysis. There have been many, many done, dozens along the way. So what about some of the empirical research that incorporates brain measures? And the brain measures we've used over the years, and I'll focus pretty much on MRI and fMRI for structure and function, but we've used MRI and fMRI, EEG, ERPs, near-infrared, event-related optical signals, eye tracking, and a variety of other things. So let me show you some of the data. So again, we need some conditions in these randomized control trials. And Mr. or Ms. Centipede is pretty sure that walking is the best exercise. I don't know if it's the best, but it's certainly an aerobic exercise. So that tends to be one condition. Most of these studies are focused on aerobic exercise, things like swimming, jogging, bicycling, but more recently on Eastern practices such as Tai Chi and yoga, as well as resistance exercise or weightlifting. And they all show benefits. Which is larger or smaller, it's hard to say. There aren't a lot of systematic comparisons, but most of the research, because it's scaled up from the animal work, is focused on aerobic. And we actually know a lot more about the underlying molecular and cellular changes of aerobic exercise. Mr. Gumby didn't do very well in his toning and stretching, and that tends to be the control condition. It's another form of exercise, but it is not aerobic, and you don't see improvements in cardiorespiratory fitness. And I think one of the hardest things to do in many of the randomized controlled trials we do in neuroscience and neuroergonomics is get a really good control condition. Because when I think of cognitive training, and you're training, let's say, memory, it's pretty hard to get a control condition that's just as engaging, but doesn't incorporate any memory. That's a tough one. And this is why I think cognitive training has been so controversial. I also work in that field, but I'm not going to tell you about that today for the most part. So see, these are some data we published a number of years ago now. These are the first data I know of, and there have been many, many replications of this simple study. These data represent a double subtraction. That is, after 6 months of exercise, 3 days a week, an hour a day, two conditions: walking, these are older adults over 60 years of age, up to 85 years of age, and the control is toning, stretching, and light strengthening with the rubber bands, you see. And what we've looked at, what we have here is the difference between the walkers and the toners, that is the condition we're interested in, the aerobic condition, and the non-aerobic, in terms of changes in brain volume with a technique that's not used so much anymore. They're much better techniques called voxel-based morphometry. And the little blue dots are increases in gray matter in the individuals who walked across 6 months, 3 days a week. Nobody's winning any races here. They're simply walking more. We tend to work with when we work with older adults, for the most part, not always, but for the most part, it's pretty sedentary older adults. And it's pretty easy to get them, unfortunately. The little yellow patch in the middle and the right-hand slice is actually in the anterior commissure, and that's increased white matter volume. It's in the GNU of the anterior commissure, and then we'll come back to that again a little later. So these are structural data, the first I know of. There have been many, many replications of this. So we can actually drill down a little bit further to particular nuclei. And we know from the animal research that neurogenesis occurs in the dentate gyrus of the hippocampus. And we can look at that, and we did here. And this is a year-long study, again, 3 days a week, sedentary walkers versus toners. The blue line are the walkers in each of the panels. The red line are the toners, so our active control condition. And what we see in the left and right hippocampi, again, this is purely structural, is an increase in the volume of the hippocampus, and the average age here is about 70 years of age for the walkers, but a decrease, which is pretty typical over time for the toners. That is, volume of the hippocampus and many nuclei does decrease. And we have a couple of control nuclei here that really don't show much of a change. Now, I'm not saying this is due to neurogenesis. We wouldn't know from human data, But that's one of the possibilities. Of course, there are other possibilities like vascular changes, as well as synaptogenesis, nerve growth factors, and so forth. But this suggests that there's a carryover, a consistency between the animal research and the human research in terms of the kinds of changes we see. We can also look at white matter tracks using a technique called diffusion tensor imaging. It's one of many we can use. And what we have here is an analysis. The 4 panels on the left-hand side are analyses of different regions of the white matter tracks from the genu, which is in the front of the corpus callosum, to the body of the corpus callosum, to other regions as a function of VO2 max. Now, VO2 max is the gold standard for cardiorespiratory fitness, for measuring cardiorespiratory fitness. It's really pretty simple. You get on a treadmill, you walk and sometimes jog faster, and sometimes it gets steeper. You have an oxygen mask on with EKG leads on your chest. So far, this is the gold standard. You don't have to use this. It's great when you can. It's good to have a cardiologist sitting there, especially if you have older or high-risk people, because negative things can, of course, happen. I'm trying to get rid of the poll here. Let's see if I can do that. So there are changes that relate, think of these as dose response effects that relate VO2 to the integrity of the white matter. Okay. How about brain function? What are the changes? Well, there are many ways to look at this, from particular regions of interest to functional connectivity. So I'll just tell you a little bit about functional connectivity. And we know from the bottom relationship that aerobic fitness is related to cognition. The question we asked here is whether functional connectivity of networks, and these are some networks defined by Yao. He's defined 7 and 14 of them, and other people have defined other networks. And what we're going to do is look at this relationship. It's pretty easy to map out these networks because there's an awful lot of literature that suggests. It didn't look like people were too good at exercising here. Not bad. Okay, the neuroergonomics folks aren't too bad. So you wouldn't be my most sedentary anyway. So this is color coded in terms of different networks, from the default mode to the frontal executive to the frontal parietal. We can look at many networks, and we have. These are data that are interesting. These are simply cross-sectional data. We characterized 190 older folks and 26 younger folks to essentially map the networks individually in everyone. And these are some of the networks illustrated by yellow for default mode and dorsal attention for red, which is more of an attention network, and other networks here. And then we asked a simple question in this cross-sectional study. Well, first we asked whether the interconnections within and between networks was different for young and old. And on the top left, we see that young have many more connections within networks than old. That is, people have talked about this, researchers have talked about this as de-differentiation, because if we look at the bottom panel, old have connections more than young, and they tend to be between, not within networks. So the brain tends to be less specialized. And then we ask the question, so what happens? So there are 3 bars for each color. The networks are on the bottom here. And we ask the question of whether high fit elderly, and that would be the middle bar in each color, individuals from 65 to 85 years of age, look closer to the young than they do the low fit elderly. And we see that in particular if we look in the middle panel with the 3 larger bars, the yellow bars, default mode core regions. We see that the high fit elderly look more like the younger individuals than the older low fit. And this is in terms of network connectivity. We'll get back to this in a second. These data, which are a result of many, many terabytes that we've collected in a randomized controlled trial with 120 participants, simply older people, 2 groups. Well, not really simply older, but we'll get to that. One group, represented by the blue bars, are the individuals in a year-long program, 3 days a week, in our toning and stretching condition. And the yellowish orange bars are the walkers. And we can focus on any one of these panels. Let's take the top left. These are connections in the default mode network. And what we see is for the blue bars, they go up and then they go down a little bit. They really don't go anywhere over the course of this year-long exercise program. But the yellowish orange bars continue to go up. That's interesting, and that's consistent with the cross-sectional data. Remember, these are longitudinal data, so we can infer causality from them. But also interesting here is the unfilled white bar. Those are the young adults. And what we see across all these panels is that the older high fit, the older people who become more fit, and not high fit, but become more fit across the year, become more like the younger adults. That in and of itself is interesting because that suggests that dedifferentiation isn't inevitable. So what about the relationship between changes in connectivity as a function of exercise and changes in cognition? So we created a few latent variables with multiple measures of different constructs. And what we see here are modest correlations for different regions in the default mode network, illustrated on the 3 on the left, and executive function, things like multitasking, working memory, cognitive flexibility, and so forth. So these improvements in connectivity within network are related to improvements in executive function. So there is a functional significance then for improving physical activity and exercise. So we can put a little check mark then on the right-hand side here that there is this relationship between aerobic and fitness and cognition that's partially mediated by functional connectivity. We also take other measures. We draw blood, more so from older folks than kids. Kids have no problem, even young kids going into an MRI machine. They have a lot of problem if you stick a hypodermic in their arm. They don't like that too much. But older folks, speaking as an older person myself, I am a senior citizen, we get stuck all the time. It's not a big deal. And what we see here on the left-hand side is our connectivity, one panel of our connectivity results. And what we see on the right-hand side on the top is our non-aerobic or toning and stretching group. We're relating BDNF change, brain-derived neurotrophin factor, from plasma that we can extract from blood to connectivity change. There's no significant relationship. We move down to the bottom, and there is a significant relationship, much like in the animal research, but in the animal research, BDNF would be extracted from the hippocampus, from a hippocampal biopsy. None of our older people ever volunteered for a hippocampal biopsy, so the best we're going to do is blood. But there still is this relationship. These are some data from my colleagues and I, Lars Nyberg, and others at the University of Umeå in Sweden. And what we look at here is D2 dopamine receptor availability. There's a lot of work, again, on neurotransmitter changes in animals. This is tougher to do with humans, but this is using positron emission tomography and a particular ligand that's sensitive to D2 or dopamine receptor availability. And what we see, we can look at any of these panels, but let's look at the top right one. We see a relationship between the DRD2 and aerobic fitness. So again, trying to bring the molecular and cellular research closer to the human cognitive or neuroergonomic research. Another question we might ask is whether we can use some of the neuroscience data in a predictive sense. That is, can we predict based on characteristics of individuals, be they behavioral, psychosocial, or neurophysiological or anatomical, some of the potential training benefits. And we've done this in a number of ways, and then others have too. And I'll tell you about Michelle Voss and just a bottom line of what she found, which is really interesting, in a second. But these are some data that we published in collaboration with a researcher by the name of Mark Desposito at Berkeley and a former student of mine, Pauline Bankert, who's done a postdoc with Mark in his lab. And we used a technique called modularity. And modularity, simply defined, is the extent to which within network connections are stronger than between network connections. And this can be scaled in a ratio manner. And there is a good deal of data that suggests that brain modularity predicts cognitive training benefits up until this study, cognitive training benefits in older individuals, in younger individuals. There's now a paper out with children, with individuals with traumatic brain injury in terms of recovery from TBI. And what we looked at here was whether this metric, this within network connectivity, essentially, modularity scaled in a ratio sense, predicts who's going to benefit to the greatest extent from exercise interventions. So the data are collected in the first session before there's any training. Then we do the exercise training. for either 6 months or a year, and we try to predict who will improve the most, particularly who will improve the most in cognition based on the brain data. So let me show you a little bit about the data. So this was a 6-month exercise intervention, 4 different groups. I'll show you what they were in a second, over 100 participants. The groups were walking with a nutritional supplement that had something in it called beta-alanine, which helps you build lean muscle mass. So it's an indirect way to increase exercise. Dance, this was social dance, and toning and stretching, that's the SSS. So those are measures, they're relatively modest, very modest changes in cardiorespiratory fitness. Anything above the 0 line here are improvements in executive function across the 6-month period. And we found those improvements for 3 out of 4 of the groups. And here's the bottom line. And the bottom line on the left-hand side of each of these panels is executive function gain. Again, this is at the latent variable level, a concatenation of multiple tasks of executive function. And on the x-axis is modularity. So what we find across the board, which is interesting, across these different interventions that improve physical activity and to some sense improve the cardiorespiratory fitness, we see that brain modularity at baseline does predict the magnitude of the benefit across these intervention types. So this is true with brain modularity as a predictor, not just with cognitive training, but also with physical training. This is just the bottom line. I pulled out some slides to make this a little briefer, but let me tell you the story, and then you can find Michelle Voss's paper. It came out last year. And what Michelle has done is simply asked another question about predictability, and that's at the top here. Can acute exercise, single bout exercise, and the brain changes that occur from either pretty minimal or a more modest level of intensity predict the benefits in terms of cognition and brain connectivity of a chronic exercise program, that is from 20 minutes to 12 weeks. And Michelle looked at 3 different networks because these are networks that are relevant to a lot of the animal research on exercise and memory, posterior medial, anterior temporal, and ventrolateral prefrontal. And any of the lines here, especially as they get thicker, predict from these networks, that is the benefits or the differences in brain activation found for the acute exercise bout, again, 20 minutes, actually predicts the benefits both cognitively in terms of brain connectivity for the chronic or 12-week program. So you can use brain modularity, you can use acute to predict chronic. There are a few ways to actually predict benefit. Of course, the next step might be, so how do we change brain modularity independently of other things and boost it so people will get more out of the interventions? And how do we take account of these data with different memory networks to both boost benefits and predict benefits? So we've also developed over the years, I'll just tell you about a couple of them, a number of new tools for studying fitness, brain, and cognition. Some of them are extremely simple, some of them not so much, and I'll just tell you about 2. One of them is very simply, we usually look at mean, median, or mode effects. ROIs, connectivity, or what have you. Sometimes we use machine learning and artificial intelligence. There are many different ways to do this. Things like multi-voxel pattern analysis with support vector machines. We've done a bit of that, and we are doing a bit of that. But here, this is very simple. It's simply saying what regions of variability in the BOLD or fMRI signal relate to performance on fluid intelligence and memory, and any of the little brain slices here with blue patches relate for variability, not for mean effects. The mean effects are subtracted out. These are independent effects to fluid intelligence and memory. And then we take these regions as masks and we superimpose them on fitness effects. And we ask, so how much of this could be due to fitness? And the answer is quite a bit, fitness or physical activity. And we divide the physical activity into light, LI/PA versus moderate to vigorous. So we can use variability to actually reveal residual variance effects between cognition, fitness, and physical activity. Residual as compared to means or averages. We also, and this is in collaboration with Curtis Johnson and a number of other folks at the University of Illinois, Curtis and Brad Sutton, our bioengineers, have come up with a measure of hippocampal viscoelasticity. Think of it as plasticity of brain tissue, be it gray or white matter. And this is interesting. People have been looking at plasticity or mechanical properties of brain tissue for years, but mostly at the single unit or multiple unit level. The question is, can we look at it with massive numbers of cells, as we do with human measures of MRI and fMRI? And the answer is we can. And as we see from this mediation diagram, again, at the bottom, we know cardiovascular fitness is related to cognition and memory in particular. And what we found is the differences in hippocampal viscoelasticity is a moderator of part of this variance, and in fact accounts for more variance than simple measures of volume. So another measure that can be used to reveal intervention-based changes in brain and behavior. We're often asked, what about, is there a point of no return? That is, do some people not benefit from these interventions because the brains have just gone too far in the wrong direction? Perhaps they no longer have the plasticity. And I could show you many slides, but I won't. I think I'll just show you one. Let's see. Skip the next one. But there are a number of researchers. Pat Henn in the top left here is a researcher at the University of Colorado in Denver. And she's found the little bar in the middle suggests that the effect size for early AD patients of exercise is just about the average effect size, in fact, a little larger than it is for what we call neurotypical older adults, that is folks who don't have Alzheimer's or age-associated neurodegenerative diseases. That is, even Alzheimer's patients and mild cognitive impairment patients can benefit in terms of cognition from exercise. Bottom left is some data from Ergen Uck's lab. He's the head of the Alzheimer's clinic at the University of Iowa and the VA center there. And Ergen's looked at both brain changes using fMRI in a Erickson flanker test, but also changes in attention of Parkinson's patients who are getting exercise. And these patients tend to look more like younger patients, and their attention measures tend to improve in the flanker paradigm. Ruchika Prakash on the right-hand side is a researcher in the medical school and in psychology at Ohio State University. And the two brain slices on the left indicate that MS patients tend to have greater gray matter, those are the yellow patches, if they're involved in exercise, usually on stationary bicycles, because they tend to be quite unsteady, as the Parkinson's patients are. And the brain slices on the right, these are smaller effects, but these are white matter patches from diffusion tensor imaging. And of course, MS is a white matter disease. And what these data suggest is that those individuals with multiple sclerosis who are exercising tend to have more integrity in their white matter. And it tends to relate to better perceptual speed. I'm going to skip this one. That's cancer patients, interesting story there. But what about fitness effects across the lifespan? So I've told you mostly about older folks and then patient groups. Let's start on the top left here. And if we start on the top left, another simple moderator mediator design. And again, we're looking at memory on the right, and in fact, a kind of memory the hippocampus loves to help out with, and that is relational, declarative, or episodic memory, relating different pieces of information. And we ask the simple question, this is with 7 to 10-year-olds, as to whether hippocampal volume is important in this relationship. If you look at the top right, this is an average between the left and right hippocampi for the lower fit and the higher fit. These are cross-sectional data. Kids, and we find that kids that are higher fit tend to have larger hippocampi. We look at the bottom left, and we can see two different tasks. One of them is an item memory task on the left-hand side. The other one is a relational memory task. The hippocampus, of course, participates to a greater extent with relational memory, and that's where you see the difference in performance between the high fit, the dark bars, and the low fit, the gray bars. And we see a dose response effect between hippocampal volume that's related to aerobic fitness and physical activity and relational memory accuracy across the kids represented here. So I wouldn't say the kids are just like old adults in terms of the effects, but children also we also find benefits for kids, both normal weight kids and in collaboration with folks at the University of Granada in Spain with obese children. Let's see if I can get this to run. I don't know. I don't even see how I can click it here. Okay, let's just pretend. So I won't click it. This is a VR setup. I've been doing VR for more than a few years. And this is a completely immersive VR. There's a little kiddie treadmill there. And if you could see this, you would see a situation in which the little girl, she's an 8-year-old girl, is asked to cross the street. And she'd either be asked, and we create the traffic. So we're allowed to run over children here, which you're not allowed to do in the real world, of course, right? You're not allowed to run over kids. And we do, we run them over all the time because we make it challenging. And we ask the question of whether the kids under different conditions, paying attention, they wear stereo goggles, so they can see everything in stereo, whether the kids that are high fit do better than the kids that are low fit, in anticipating when the vehicles will be coming at them and therefore either crossing or not crossing. Interestingly, high fit and low fit kids don't move at different speeds. Even older folks don't move at different speeds. In fact, older folks move really quickly because they're not as familiar with the VR as kids are, and tends to be quite scary. But let me show you some of the data. We have the undistracted condition on the left. That's just paying attention to the traffic, looking left and right, as you all tell your children to do. Unfortunately, they don't often do that. And there's a difference. The lower fit kids, crossing success rate, you can think of that as, do you live or do you die? Do you get run over? I can tell you, speaking as somebody who's been run over in the city of Boston once, crossing the street, it's no fun to get run over. So none of us want to do this. We go to them listening to music. And again, we see a slight difference between the high fit kids and low fit kids. But where we really see a huge difference is between when the kids are using a hands-free Bluetooth cell phone. And I don't know about Europe, the Europeans can tell me, or Asia, and the Asians can tell me, but in the United States, when we legislate against using a cell phone and driving or walking, it's a handheld cell phone. It frankly doesn't make any difference. Accident rates tend to be about 400% higher if you're using a cell phone, be it handheld or hands-free. And what we see here is the high fit kids are better able at negotiating these multitasking effects and not getting run over. That is the solid black bar in the multitasking situation than the low fit kids. So what have I told you about? And then briefly follow up with one last experiment or a little data. That this is a very, very fast tour of the literature. There are a number of literature reviews, some of them quantitative meta-analyses and so forth that you can look at, and I'd be happy to recommend. That a variety of perceptual and cognitive abilities benefit from relatively short-term programs of exercise. We see increases in brain volume in regions that normally show age-related decline, including the hippocampus, and we see developmental milestones being reached quicker for kids. Kids don't always have better connectivity or more volume. They go through periods of increases and decreases. Changes in functional brain networks, often in the direction of younger adults for older adults and older kids for younger kids. Promising fitness and brain effects with children. There's less research there, but more and more all the time. And what I didn't tell you about today, these projects do fit into the social neuroergonomics group because we look at anxiety, depression, self-esteem, and self-efficacy, and we see benefits there too. Slightly different story, but benefits. But you know, there are always more questions for the students attending the presentations here. There's always plenty to do, and we've learned a lot and we don't know a lot. We've learned many things we don't know. The first point, understanding boundary conditions, and I cite a study here. This is a pretty big study, lots of subjects. The main goal was not cognition, and it was individuals who were frail and mobility challenged, but they didn't find any benefits from exercise. Why is that? What are the boundary conditions? We also want to know more about combining different interventions, because in the real world, it's not just exercise or social interaction or cognitive activity or diet. It's all of the above in interesting ways. And we and others are starting to look at that. Genetic moderators, that's a tough one. Doing GWAS and looking across the genome requires lots of participants looking at single genes, such as APOE, a gene related to Alzheimer's dementia, and others account for very little variance, but we and others have done some of that. And then beyond the laboratory door, the little panel on the bottom right is from a colleague in Georgia, and they find, and we find, that with kids, that not only do kids do better on executive function and memory tasks if they get more fit or are more fit, they do better on school achievement, which most parents would be very happy with. And then finally, here's some data from a study that a number of us did together at the Beckman Institute a few years ago. And it was a study funded by, most of our funding comes from the National Institutes of Health. This is from IARPA, the Intelligence Advanced Research Projects Agency. They asked us a simple question. How do you make people smarter? Maybe not such a simple question. So we and a few other groups pursued that. And here's the way we tried to make them smarter. We did a few experiments with a few hundred young to middle-aged people with cognitive training. I developed that with a student of mine, brain stimulation on the dorsolateral prefrontal cortex, physical fitness, and in another study, we actually used some meditation or mindfulness training. And to summarize a tremendous amount of data, let me show you this figure or table. On the left-hand side, we have the metric. Again, we're looking at adaptive reasoning or problem solving or decision making. That would be related to things like fluid intelligence. In the middle, we look at the nature of the intervention. One column over, Cohen's D, that's the effect size, and then the very specific measure. And what we see if we go down the intervention column is that fitness plays a role in all of these combinations that give us anywhere from intermediate to fairly large effect sizes of intervention training on decision-making, different metrics. Other things contribute, like Mind Frontiers, is the cognitive task. It's a gamified, multimodal, multi-aspect of cognitive task that we develop. And meditation, Wales meditation. There are many different varieties. But what we see here is that fitness plays a role, or physical activity. These are 4-month programs with 18 to 44-year-olds across the board. So we've started, and others have started, the finger study and others in looking at multimodal kinds of interventions that represent the real world and life a lot more. That's it. If there are any questions, I'd be happy to try to address them.